Hi, this is Michael Edwards, Assistant Professor of Medicine at UC Denver, specializing in bioinformatics. This is part 5 of the IPA tutorial. We will talk about upstream analysis and regulatory effects. Our previous tutorials looked at groups of genes, interactions between groups of genes. So now we're going to take all of those genes, basically everything on our gene list, and try to find overrepresented targets, not only of upstream regulators such as genes, but of chemicals and microRNA. This is a very useful tool, and I would say, in my opinion, it's one of the most valuable tabs on IPA. Um, so we're on the Upstream Analysis tab. Given here are the upstream regulators that have overrepresented targets on our gene list. That means that we are selecting these targets more than we would expect by random chance. Given here in this first one is the BCR complex. This is B cell receptor complex, I believe. And again, you can click on any of these and get more information on them. Yep, please run. B cell receptor complex. Canonical pathways, what it regulates, what it's regulated by. We saw this in the networks. We saw ERK. I actually did see seq in one of those. And doing my own reading, and obviously if this is your own study, you're going to know your system. The paper or where this data set came from basically mentioned this BCR pathway and said it was more active in the activated B-cell lymphomas, or the more aggressive. And sure enough, as we're showing here, it's predicted to be more active. This is the activation Z-score. This column here notes this bias basically says that given our original gene list had more upstream than downstream, or downstream than upstream, so there is actually a bias as far as this activation. It usually holds no matter what your gene list, but this is the p-value, so these are the odds that we would get all of these 27 molecules associated with this BCR complex in our gene list by random chance. 1.2 times 10 to the negative 17th. So we can look at this more closely. So I can click on this and say display as a network. And so here is the BCR pathway. Here are all the genes associated with it that were on our gene list. We can overlay p-values and log ratios. These are all the genes. If there's an orange line, so dashed lines is an indirect relationship. A solid line is a direct relationship. Orange lines signify that the BCR complex it agrees with whatever the software predicted its activation state. So that this orange line here, BCR, is known to induce, there's BCL2 again. To induce BCL2, and sure enough, it's going up. All those genes in red are going up in the more aggressive tumors. All those in green are going down. The only ones that are not following this pathway are these genes in the yellow. The evidence for this complex being more active outweighs statistically the evidence that there's nothing going on or that it's inhibited. Close this window. And this is a really interesting function here. So this is the Mechanistic Networks. So what it does, and I want to mention again, we do not measure BCR mRNA directly. We just see its effect in the transcriptome. That all of these genes that are different contain more than you would expect for the BCL receptor or affected by it. What we can also do is now take all these upstream regulators that now we can presume that they potentially might be active in our system and start making networks out of these. And this is what this column does. So this first number here says how many target genes were used in order to make this network. And these were the upstream regulators in this column that were used for to construct the signaling pathway. We can click on this. So this is almost the crib notes of what's going on here. We got BCR complex interacting with everything in orange is predicted to be more active, and it's all based on all of these target genes, and there's a ton of them. We can click here. So here are all our target genes, the full change, what it does, and then whether it's a target of any of these molecules here in our pathway. BCL6 is predicted to be down. If you understand this, for those of you that know cancer biology, this makes sense that you would predict this. Again, if you look at the original paper, STAT3 is predicted to be more active in this system or in these aggressive B-cell lymphomas, and sure enough, it's predicted to be going up. And here are all the genes that show that. So if I click here and select this, these are all the target genes given in alphabetical order. Here's its fold change or log ratio, and then in the data set, this is what it's known to do, so that when CD9 is going up, it's predicted to be more activated, or STAT3 is predicted to be more activated. And we can click on any of these relationships to find out what are the target genes that are predicting this. Okay. Again, I think this is an incredibly useful page. A lot of times, especially with cancer genome, what you're looking for are things that don't make sense. And if we go down here, so you'll see CD38, this molecule here. In our gene list, the transcript levels actually go down. But when we actually look at its target molecules, it has more than the expected number, and it's predicted to be more activated. Now, in cancer, if everything was working correctly, the cell wouldn't grow. There would be checkpoints. So, I mean, these are the things you kind of look for in these type of diagrams. Here's the mechanistic network for it. And where is it? It's CD38 right up on top. So again, even its downstream or upstream regulators that it interacts with also are predicted to be going up. So something's going on. Even though we're trying to suppress this, and if you actually look this up, this makes sense as far as being related to B-cell lymphoma. So that even though its expression is repressed, for some reason its activity is going up, and that's exactly what you look for. We can make a network out of both of these and see potential common targets downstream. Display as a network. So here are all the targets on our gene list. Red means they're going up in the more aggressive type. Green means they're going down. And again, you can see that they do share some similar genes, or at least targets. Okay. You can sort this any way you'd like. You can sort it by activation Z-score. So right now it's scored by the significance of the p-value, or the odds that you would get all these target genes by random chance. We can sort by p-value. So these are the molecules that are predicted to be inhibited. A lot of these, so these are kinase inhibitors, and they're typically associated with cell growth, apoptosis, potential use for a chemotherapy against this, and these are all being inhibited. LET7 is a well-known inhibitor of lung cancer formation as well as other cancers, and this is predicted to be going down. So again, we got a drug, and we also have microRNAs. And if we go down here, we can see some genes. And again, some of these genes that are being inhibited, I saw a lot of kinase inhibitors down here. Things that you would expect. If you want uncontrolled cell growth, you're going to inhibit these. We can sort it the other way. These are things that are predicted to go up. We're seeing a lot of immune type inflammatory responses, and NF-kappa B predicted to go up. Again, STAT3 Here it says the log ratio. We're actually measuring it going up, and it's also predicted to be going up. There are 34 targets of stat 3 in our data set. We can look at all of those. Oh, and we also have those as well. I could select any of these and make its own gene list and analyze those alone. Okay. This is a very important page, and I can't stress enough that, it kind of goes to the theory that important cellular processes, in these tissues in your system, you're not going to wait around for it to be made. You want something there right, to have pinpoint control of your system. Things like P53, you very rarely see it change at the transcript level. What happens, what alters its activity is phosphorylation, removing a inhibitor, translocating it to the nucleus. Things that are invisible in the transcriptome. You also have things that are secreted by other tissues that you would never see. This allows you to attempt to get at this. And I would say that IPA is one of the few software systems that really exploit this type of analysis. And I think it's very, very important. This is almost like the skeleton of your transcriptome. You can go to regulatory effects. So in this page, what they're actually doing is combining these upstream regulators with these target genes, and then looking to see if they're connected to common biological diseases and functions. So here, these are the regulators that are used. There are 12 of them. So these are the upstream regulators that we might not be measuring directly, but we believe are associated with our study. The number of target genes, here are the target genes for those. So this is what we're using in our data set that are common targets for all of these upstream regulators. And these are the disease or functions that are common between those. And we can click on this. And this is nice. I mean, this is what you're looking for. You're looking for things that you wouldn't expect to be tied to say an upstream regulator and also shared with a biological function. So you're almost using two biological pieces of information to not, you can find gene lists or relevant target or upstream regulators. And these are all given here. So again, we don't measure these directly. We just know that we are guessing that they're probably active in our system based on the fact that their downstream targets are overrepresented on our gene list. And not only that, if we go down here, we can see a lot of these biological functions that they're associated with. And it doesn't take too much of a stretch of the imagination to see it. You have apoptosis going down and lots of cell growth. Very much indicative of an aggressive type of cancer. And you can do this to any of these networks. All right, I'm going to stop there. Thank you very much. I think in the next tutorial, what we'll look at is, how can you construct this from scratch using just your gene list? And this will be the new my pathway. So you're basically constructing your own pathways based on your preferences. All right, thank you very much.